Good evening, everybody. We need no reminder that we live in a world filled with pathogens, including viruses, bacteria, and numerous other microorganisms. We're continually exposed to these organisms. Yet, as a species, we do an amazing job of fighting off infection. So how do we persist in the face of this diverse, evolving set of pathogens? We do this by means of an immune system. So we have this intricate, amazing immune system that allows us to fight off infections by means of cellular players. These cells identify and respond to molecular signatures of pathogens, like the 4 T cells colored in red, attacking the infected cell in the image on the right. So we have adaptive immunity. This is our ability to have targeted immune response against specific pathogens, along with memory. Okay? This is how vaccines work. So the key players are T cells and B cells. T cells can directly kill infected cells, while B cells are the cells that make antibodies, which are molecules that can help clear infection as well. So both T cells and B cells physically engage other cells. They do this using surface receptors, denoted in this image by the T cell receptor, or TCR, and the B cell receptor, or BCR. Okay? The fact that these cells physically contact each other means that there are forces on cells. So they literally push and pull on one another. So a central unresolved question in immunology is, how do immune cells identify molecular signatures of pathogens and distinguish between foreign, that from the pathogen, and self, that from our own bodies? An emerging paradigm in immunology is that forces are an important regulator of immune cells. So while that's the case, there's a gap. And that gap is that we need a framework for understanding the role of forces in immunology. So that's where my research group comes in. So we use mathematical modeling and computer simulations built on physical principles to understand biology. This allows us to provide insight that's inaccessible to experiments alone. We use theory rooted in the physical sciences. So part of my group studies mechanical forces at immune cell interfaces. We've worked on a variety of problems ranging from T cells to B cells. We've studied T cell receptors, scanning protrusions from cells. And today I'm going to tell you a bit about a project that looks at the interface between a B cell, the cell that produces antibodies, and antigen-presenting cells. Okay, so the image on the right shows a B cell on top interacting with what's known as an antigen-presenting surface or antigen-presenting cell. So an antigen is any substance that will elicit an immune response. So you see it has B cell receptors, or BCRs, that interact with the antigens, and the B cell generates forces within the cell to help distinguish foreign from self. So in a cartoon picture, what you see is the B cell receptor, or BCR, interacts with these red antigens. There's a force depicted by this arrow on the right that sort of pulls the membrane inward, eventually leading to a large invagination and eventually a complete internalization of the antigens, which is part of the immune presentation process. So I'm going to tell you a bit about our mathematical modeling, which we do in my group. So we focus on simple models that capture key physical features of cells that allow us to gain physical insight into how they work. Okay, so we capture the key features like randomness, forces, large-scale deformations, and we use computer simulations to understand the consequences of these effects. So what you're seeing here is a result of one of our computer simulations. There's a B cell surface represented mathematically on the bottom, an antigen-presenting surface on the top, and you see these red balls emerge. These are the B cell receptors that are binding to antigens on the other surface. And so what you see here is this sort of transient deformation followed by flattening of these membranes. This is representing the process when the B cell receptors bind to the other cell surface. So now what we're seeing is now these B cell receptors are interacting with a foreign antigen, something from a pathogen, where they interact very strongly, can actually deform the membrane. So our mathematical model is capturing some of the key features that you would see at a B cell interface. So how do B cells discriminate between self and foreign, or between different antigens? Well, they do this by having sharp thresholds. And so this axis on the right, I also like charts, by the way, and figures. So this is what's known as affinity, or how strong you bind to something. B cells tend to bind more strongly to things from outside, from foreign molecules. And so what you see then is, as the affinity, or the strength of binding, goes from small to large, if there's no forces, you see this gradual response, which means that you can't really distinguish between things very well. In contrast, when you have forces, or this mechanical feature, you see a sharp discrimination where you go from low to high over a very narrow range. This is what you need to have a binary decision where you can decide one versus the other. Now what happens is, as you stiffen the membrane, which is what happens as a B cell matures, you actually shift the location of this of the threshold, which means that you can distinguish between different affinities. So B cells actually undergo an evolutionary process in our bodies to generate antibodies with higher binding affinity. This is part of that process. So what's the emerging picture that we have from this sort of underlying mathematical and computational theory? Well, the idea is that cells generate forces. These forces provide a test of binding strength, which is a proxy for whether it's from something foreign or from ourselves. And there's a critical event where you can see in this depicted here by these red circles, you need a cluster of these B cell receptors to bind. Once they do that, what you find here in this figure is that as you increase the number, the force that each one of these bonds experiences goes down because they share the load of deforming the membrane. Okay? However, stiffer membranes still require larger forces to deform, and hence you see this stiffness-dependent phenomenon. Okay, so what do we have here? We have this emerging picture of mechanical forces in immunology. Okay, so my group is providing the mathematical sort of underpinnings of this idea. So traditionally, people think about sharp thresholds as being a consequence of things happening inside of the cell. What we're proposing is it's actually things at the surface of the cell, the forces of the mechanics, that can influence cellular responses. So in the future, we're really interested in having a fundamental understanding of immune cells, along with applications in the engineering of immune cells. For example, in cancer immunotherapy, understanding the effects of tumor microenvironment where stiffnesses can vary, and understanding how to activate cells in different contexts. With that, I would like to acknowledge my group. So particularly the work of Dr. Bing Li in the lower left there in the plaid. He was a former postdoc in my group, along with Rob Pullen, who is next to me. I'm the one on the far right. He's the one next to me. He's a former PhD student. And I'd like to acknowledge funding from the National Science Foundation. Thank you.